हेलो एवरीवन, वंस अगेन वेलकम बैक टू अवर चैनल बीट द नीट। एंड कैसे हैं बच्चों आप सब लोग। एंड आई होप कि आपकी तैयारी बहुत बढ़िया चल रही होगी, क्योंकि बहुत कम टाइम रह गया है नीट एग्जाम के लिए। एंड आई होप कि आप लोग इस शॉर्ट जो आप लोगों का क्विक रिवीजन पॉडकास्ट सीरीज मैंने स्टार्ट की है, एंड इससे आप अपना रिवीजन अच्छे से कर पा रहे होंगे। इसको जो एक अच्छा सा मैं तरीका आपको बता रही हूँ, जो और बच्चों ने शेयर किया है, कि जिस तरीके से अगर आप करेंगे तो आपको मैक्सिमम बेनिफिट मिलेगा। जब आपको ये चैप्टर अच्छे से प्रिपेयर हो जाता है ना, और आप उसके बाद में आपको उसको दोबारा से 2-3 दिन में फिर से रिवाइज करने वाला सीन जब आपके साथ होता है ना, तो उस टाइम पे अगर आपको उस तरह से रिवाइज करने का टाइम नहीं मिल रहा है, तो आप अगर इन पॉडकास्ट सीरीज से उसको सुनेंगे तो आपके जितने भी इंपॉर्टेंट पॉइंट्स, इंपॉर्टेंट टर्म्स, जो भी कुछ है चैप्टर में पूरा इंपॉर्टेंट जो आपके नीट एग्जाम में आने वाला है, तो अगर आप उस तरीके से उसको सुन करके रिवाइज करेंगे तो आपका बहुत कम टाइम में कई सारे चैप्टर्स आप रिवाइज कर लेंगे। एंड बिलीव मी, लास्ट तक ये सिचुएशन आ जाएगी कि आप आराम से पूरी बायो एक दिन में रिवाइज कर लेंगे। एंड आई होप कि आप लोग इसको अपने दोस्तों के साथ में भी शेयर कर रहे होंगे, ताकि उनको भी इसका बेनिफिट मिले। सो आज हम करने वाले हैं क्लास 12 का मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस। थोड़ा लंबा चैप्टर है, थोड़ा सा ये वीडियो लंबा होगा, बट सारे इंपॉर्टेंट पॉइंट्स कवर होंगे, क्योंकि बच्चों ने मुझसे बोला है कि मैम वीडियो थोड़ा सा लंबा भी हो जाए तो भी कोई बात नहीं, बट आप सारे पॉइंट्स प्लीज़ कवर करिएगा, कोई भी लाइन मिस मत करिएगा। तो भाई मैं उसको ज़रूर फॉलो करूँगी, और कोई भी इंपॉर्टेंट चीज़ मेरी कोशिश है कि मैं बिल्कुल भी मिस ना करूँ, क्योंकि आप अगर मुझ पर भरोसा करके इसको सुन रहे हैं तो आपका हर एक पॉइंट, हर एक लाइन रिवाइज होनी चाहिए। तो चलिए शुरू करते हैं चैप्टर मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस। एंड द फर्स्ट थिंग इज इंट्रोडक्शन। Deoxyribonucleic acid (DNA) and ribonucleic acid are two types of nucleic acid found in living system. DNA acts as the genetic material in most of the organisms. RNA through it acts as genetic material in some viruses like TMV, QB, bacteriophage, but mostly functions as a messenger, adapter, structural and catalytic molecule. अब आता है आपका next DNA. DNA is a long polymer of deoxyribonucleotides, and the length of DNA, usually defined as a number of nucleotides, is also characteristic of an organism. Now I am telling you the DNA content as per the some organisms which are given in your NCERT. So bacteriophage 174, it is having 5386 nucleotides. Bacteriophage lambda 48502 base pairs. E. coli 4.6 into 10 to the power 6 base pairs, and haploid content of human DNA 3.3 into 10 to the power 9 base pairs. Structure of polynucleotide chain. The basic unit of a polynucleotide is nucleotide, which has three components. A nitrogenous base, either purine or pyrimidine, it means adenine, guanine, or cytosine, thymine or uracil, any one of. And pentose sugar, ribose or deoxyribose, depends on which is it is RNA or DNA, and a phosphate group. Purines are same in DNA and RNA, and cytosine in common in both DNA and RNA, and uracil is present in RNA, thymine in DNA. A nitrogenous base is linked to the OH of 1' carbon pentose sugar through a N-glycosidic linkage to form a nucleoside. When a phosphate group is linked to OH of 5' carbon of a nucleoside through phosphoester linkage, a corresponding nucleotide is formed. Two nucleotides are linked through 3' to 5' phosphodiester linkage to form a dinucleotide. More nucleotides in such a manner form polynucleotide. A polymer thus formed has at one end a free phosphate moiety at 5' end of sugar, and at the other end of polymer the sugar has free OH of 3' group. अब हम देखेंगे डीएनए का डबल हेलिक्स स्ट्रक्चर। सो डीएनए इज एन एसिडिक सब्सटेंस एंड प्रेजेंट इन न्यूक्लियस, एंड सबसे पहले इसको देखा था फ्रेडरिक मिशर ने इन 1969, एंड इन्होंने ही टर्म दिया था न्यूक्लियन। एंड बेस्ड ऑन द एक्सरे डिफ्रैक्शन डेटा प्रोड्यूस्ड बाय मॉरिस विल्किंस एंड रोजलिन फ्रैंकलिन, James Watson and Francis Crick proposed a very simple but famous double helix model DNA in year 1953. One hallmark of the helix is base pairing between the two strands, based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratios between adenine, thymine, guanine and cytosine are constant and equal 1. The base pairing confers complementarity, a unique property to the polynucleotide chain. So, if each strand of parental DNA acts as a template for synthesis of a new strand, the two double-stranded daughter DNA produced would be identical to the parental DNA molecule. Now I will tell some salient features of DNA, and if you have a structure you can open in your book. Made of two polypeptide chains, and where the backbone is constituted by sugar, phosphate and bases project inside. Two chains have antipolar polarity, one chain 5' to 3' and the other is 3' to 5'. Bases in two strands are paired through hydrogen bonds, forming base pairs, adenine double bond with the thymine, and cytosine triple bond, three hydrogen bond with guanine, and adenine is having the 2 hydrogen bond with thymine. This generates approximately uniform distance between two strands, and the two chains are coiled in a right-handed fashion. Pitch of the helix is 3.4 nm, roughly 10 base pair in each turn. So distance between a base pair in a helix is approximately 0.34 nanometer. And the plane of one base pair stacks over the other in double helix, and this in additional to hydrogen bonds confers stability to the helical structure. Next we will see central dogma, and इसको propose किया था फ्रांसिस क्रिक ने. So states flow of genetic information as DNA, फिर RNA, फिर उससे प्रोटीन में. So DNA से DNA बनना replication, then DNA से mRNA बनना transcription, फिर mRNA से प्रोटीन बनना, that is the translation. In some viruses, the flow of information is in reverse direction. It means from RNA to DNA. So it is called reverse of central dogma. अब आ जाती है पैकेजिंग ऑफ DNA हेलिक्स. सो यहाँ पे आपका आता है मॉडल न्यूक्लियोसोम. सो ये स्ट्रक्चर आपके NCERT में मौजूद है एंड इसके सारे पॉइंट्स मैं कवर कर रही हूँ. DNA double helix in a typical mammalian cell is 6.6 into 10 to the power 9 base pair. The length is approximately 2.2 meter and it means 6.6 into 10 to the power 9 base pair into 0.34 into 10 to the power minus 9 meter per base pair. Far greater than the dimension of a typical nucleus, जिसका approximately होता है size 10 to the power minus 6 meter. In prokaryotes, E. coli, like the DNA being negatively charged is held with some proteins that have positive charges in the nucleoid. The DNA's nucleoid is organized in large loops held by proteins. In eukaryotes, it is much more complex. The positively charged set of basic proteins, histones, which is rich in lysine and arginine, are organized to form a unit of 8 molecules and it is known as histone octamer. Number 2, the negatively charged DNA is wrapped around positively charged histone octamer to form a nucleosome. A typical nucleosome contains 200 base pair of DNA helix. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin. Thread-like stained bodies seen in nucleus. The nucleosome in chromatin are seen as beads on a string structure under electron microscope. It is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage to form chromosome. Packaging of chromatin at higher level needs non-histone chromosomal, it means NHC proteins. In a typical nucleus, we have two regions in chromatin, euchromatin and heterochromatin. Euchromatin के features हैं loosely packed, stains light and transcriptionally active. Heterochromatin के features होते हैं more densely packed, stains dark and transcriptionally inactive. इसके बाद आता है आपका the search for genetic material. So सबसे पहले इसमें है transformic principle. 1928 में फ्रेडरिक ग्रिफिथ in a series of experiment with Streptococcus pneumoniae witnessed a miraculous transformation in bacteria. Some of these bacteria produce smooth, shiny colonies, while others rough colony. As a strain kills mice virulent, while R strain do not develop pneumonia, but heat-killed strain are also non-virulent. When heat-killed S and live R bacteria were injected in mice, the mice died, and S strain living bacteria were found in dead mice. Griffith concluded that R strain was somehow transformed by heat-killed S strain. It must be due to transfer of genetic material, transforming principle, but the biochemical nature was not defined. Biochemical nature of transforming principle. So scientists are Oswald Avery, Colin MacLeod and Macklin McCarthy 1933 to 1944 discovered that DNA of S bacteria caused R bacteria to become transformed. As proteases and RNases did not affect transformation, but DNAs inhibit transformation. They concluded that DNA is hereditary material, but not all biologists were convinced. The genetic material is DNA. That the unequivocal proof that DNA is a genetic material came from the experiments of Alfred Hershey and Martha Chase in 1952 on bacteriophages using radioactive phosphorus 32P and sulfur 35S in separate medium with E. coli. Now the next is properties of genetic material, like DNA versus RNA we will see here. A molecule that can act as a genetic material must fulfill the following criteria. Points are like should be able to generate its replica, it means it is able to do the replication, should be stable chemically and structurally, should provide the scope for slow mutation required for evolution, and should be able to express in the form of Mendelian characters. Both RNA and DNA can direct their duplications. The DNA has two complementary strands if separated by heating can again come together when appropriate conditions are provided. 2' OH group present at every nucleotide in RNA is reactive and it makes it easily degradable. RNA is also catalytic, hence reactive. Among the two nucleic acids, DNA is a better genetic material. Presence of thymine at the place of uracil also confers additionally stability to DNA. Both DNA and RNA are able to mutate. Being unstable, RNA mutate at a faster rate. RNA can directly code for the synthesis of proteins and can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. DNA being more stable is preferred for storage of genetic information, and for transmission of genetic information RNA is better. Next आता है RNA world. RNA was the first genetic material that essential life processes like translation, metabolism, splicing evolved around RNA. RNA used to act as a genetic material as well as a catalyst, so was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that make it more stable. Now the next is replication. Watson and Crick had immediately proposed a scheme for replication of DNA while proposing the double helix structure of DNA. That is, the two strands would separate and act as a template for the synthesis of new complementary strands. After completion of replication, each DNA molecule would have one parental and one newly synthesized strand, termed as semi-conservative DNA replication. Now we will see the experimental proof. So semi-conservative DNA replication was shown first in E. coli, then in higher organisms like plants and human cells. Michelson and Stoll performed experiments in year 1958 using normal N14 nitrogen and a non-radioactive 15N isotope of nitrogen as source of NH4Cl and centrifugation in a cesium chloride. It means CSCl density gradient. The various samples were separated independently on CSCl gradient to measure the densities of DNA. E. coli divides in every 20 minutes. So there is a diagram in your NCERT that you have to open, so you will get it very well. Now you can see here the separation of DNA by centrifugation. So in the first heavy strand we are having the 15N both strands, and in the next cycle after the 20 minutes we are having 15N and DNA and 14N also over there, and that is a 50-50, and then after we are having in the generation second 14N light strand and 14N15 hybrid, that also. So Taylor and colleagues in 1958 used radioactive thymidine and Vicia faba beans they used to prove that DNA in chromosome also replicate semi-conservatively. Now the machinery and the enzymes which is used in DNA replication, and here is a diagram of a replication fork in your NCERT in which is a one strand is a 5' to 3' direction and another is the 3' to 5' direction, and that is a 3' to 5' direction strand is the template DNA or the parental strand you can see, and here you can see the newly synthesized strands in which one is the continuous and the another one is the lagging, like the Okazaki fragments you can say. And E. coli completes replication in 18 minutes. The average rate of polymerization is 2000 base pair per second. Energetically, replication is a very expensive process. Deoxyribonucleoside triphosphate, and that serve dual purpose, that act as a substrate and also provide energy for polymerization. Replication occurs within a small opening of DNA helix called the replication fork. Diagram is given in your NCERT, And DNA-dependent DNA polymerase main enzyme DNA catalyzes the polymerization process in 5' to 3' direction. So on one strand template with polarity 3' to 5', replication is continuous, while on other template with polarity 5' to 3', it is discontinuous. Discontinuously synthesized fragments are later joined by DNA ligase, and there is a definite region in E. coli DNA where replication originates is termed ORI site, that is the origin of replication. And during recombinant DNA procedures requires a vector which provide origin of replication. In eukaryotes, replication of DNA takes place at S phase of the cell cycle. Replication of DNA in cell division cycle should be highly coordinated. A failure in cell division after DNA replication results in polyploidy, that is chromosomal anomaly. Next is transcription. It is process of copying genetic information from one strand of DNA into RNA, and principle of complementarity governs transcription except adenosine forms pair with uracil instead of thymine. In transcription, only a segment of DNA and only one of the two strands is copied into RNA, otherwise one segment of DNA would be coding for two different proteins. Also, the two RNA molecules, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent translation. Transcription unit. A transcription unit in DNA primarily has three regions: a promoter, the structural gene, a terminator. And the DNA-dependent RNA polymerase catalyzes polymerization in only one direction, 5' to 3'. The strand that has polarity 3' to 5' act as a template. The other strand with polarity 5' to 3' and the sequence same as RNA except thymine at place of uracil is referred to as coding strand. Now there is a structure in NCERT of transcription unit in which there is a promoter, coding region or coding strand, and then after the terminator. The transcription start site is a promoter, and in between there is a coding strand where the structure gene, or you can say that sequences are present, and it get terminated from the terminator. Promoter is located towards 5' end upstream of a structural gene in reference to coding strand. It provides binding site for RNA polymerase, and terminator is located towards 3' end downstream of coding strands. Now the transcription unit and the gene. Genes are located on the DNA, which is functional unit of inheritance. DNA sequence coding for tRNA or RNA also define a gene. Cistron is defined as a segment of DNA coding for polypeptide, and the structural gene is monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. In eukaryote, genes are split between coding sequences or exons, which appear in mature RNA, and introns or intervening sequence. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. Regulatory sequences are defined as regulatory genes even though they do not code for any RNA or protein. Now the types of RNA and their process of transcription. A single DNA dependent RNA polymerase catalyzes transcription of all three types of RNA, that is mRNA, tRNA, rRNA in bacteria. RNA polymerase binds to promoter and initiate transcription, and it uses nucleoside triphosphates as substrate and polymerizes in a template dependent fashion following the rule of complementarity. It somehow all facilities opening of the helix and continues elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA and RNA polymerase falls off. This results in termination of transcription. RNA polymerase is only capable of catalyzing the elongation process. It associates transiently with initiation factor rho and initiation factor sigma and the termination factor rho to initiate and terminate the transcription respectively. In bacteria, mRNA does not require any processing, so transcription and translation are coupled. And in eukaryotes, there are two additional complexities. This diagram of transcription in eukaryotes you have to see from NCERT. Now there are at least 3 RNA polymerase in the nucleus in addition to RNA polymerase found in organelles and a clear-cut division of liver. Now the enzyme RNA polymerase 1 transcribes 28S, 5.8S and 18S rRNA. RNA polymerase 2 transcribes mRNA precursor, that is hnRNA, and RNA polymerase 3 transcribes 5S rRNA, tRNA and snRNAs. The primary transcript hnRNA is subjected to splicing, where introns are removed and exons are joined in a defined order. In hnRNA undergoes additional processing called capping and tailing to form mRNA. In capping, an unusual nucleotide, methyl guanosine triphosphate, is added to 5' end of hnRNA. In tailing, adenylate residues 200 to 300 are added to 3' end in a template-independent manner. Fully processed hnRNA is called mRNA, that is transported out of the nucleus for translation. The split gene arrangement represents probably an ancient feature of the genome, the presence of introns of reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. Now the next is genetic code. George Gamow suggested that the genetic code should be triplet. Chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combinations of bases. Marshall Nirenberg's cells-free system for protein synthesis finally helped the code to be deciphered. Severoka enzyme polynucleotide phosphorylase was helpful in polymerizing RNA with defined sequences in a temperature-independent manner. Enzymatic synthesis of RNA it is basically. So now there are some features of genetic code. The codon is triplet. 61 codons code for amino acids and 3 codons are stop codons. The code is degenerate, that is some amino acids are coded by more than one codon, and the codon is read on mRNA in contiguous fashion, that is there are no punctuations. The code is nearly universal. Example, UU codes for phenylalanine from bacteria to humans. Exceptions, some variations have been found in mitochondrial codons in some protozoans. AUG has dual function. It codes for methionine and act as an initiator codon. UAA, UAG, UGA are stop terminator codons. Now the next is mutation and genetic code. Insertion or deletion of one or two base changes the reading frame from the point of insertion or deletion and called frame shift mutation. Insertion or deletion of 3 or its multiple base insert or delete in one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered from that point onwards. tRNA, the adapter molecule, Francis Crick postulated the presence of an adapter molecule that would read the code and bind to specific amino acid. The tRNA, then called sRNA, was known before genetic code was postulated. tRNA has an anticodon loop that has bases complementary to the code, and it also has an amino acid acceptor end to which it binds to amino acids. tRNA are specific for each amino acid. For initiation, there is another specific tRNA that is called initiator tRNA. There are no tRNAs for stop codons. Secondary structure of tRNA looks like a claw leaf, and though the actual structure is compact molecule which looks like inverted L. Now the next is translation. Translation refers to process of polymerization of amino acids to form a polypeptide, and the order and sequence of amino acids are defined by the sequence of bases in the mRNA. In the first phase, amino acids are inactivated in the presence of ATP and linked to their cognate tRNA by a process called charging of tRNA or amino acylation of tRNA. Protein synthesis takes place on the ribosomes, and ribosome consists of structural RNAs and about 80 different proteins. It has 2 subunits. When the small subunit encounters an mRNA, the process of translation begins. There are 2 sites in the large subunit for subsequent amino acid to bind and thus be close enough to each other for the formation of a peptide bond by the catalyst 23S rRNA. In bacteria is the enzyme ribozyme. The presence of catalyst would enhance the rate of peptide bond formation. A translational unit in mRNA is flanked by a start codon, AUG, and the stop codon. The untranslated additional sequence on mRNA are called untranslated regions, which is known as UTRs, present at both 5' end before start codon and 3' end after stop codon. UTRs are required for efficient translation. The ribosome moves from codon to codon along the mRNA. Amino acids are added one by one and translated into polypeptide sequence at the end, and release a factor binds to stop codon, terminating translation and releasing the complete polypeptide from the ribosome. अब आप इसका जो डायग्राम NCERT में दिया हुआ है इसको देखेंगे तो आपको पूरी क्लियर हो जाएगी ये सारे पॉइंट्स कि किस तरह से हैं. Next is regulation of gene expression. So gene expression results in formation of polypeptide. It can be regulated at several levels. In eukaryotes, the regulation could be exerted at transcriptional level, formation of primary transcript, processing level, that is the regulation of splicing, transport of mRNA from nucleus to cytoplasm, that is the and then after translational level. The metabolic, physiological and environmental conditions regulate expressions of genes. Development and differentiation of embryo into adult organisms are also a result of the coordinated regulation of expression of several set of genes. In prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. Then after, next is the lac operon, and Jacob and Monod were the first to elucidate a transcriptionally regulated system, the lac operon, where the lac means lactose, a polycistronic structural gene regulated by a common promoter and regulatory gene and called operon. Lac operon consists of one regulatory gene, that is I, and three structural genes, Z, Y, and A. And I gene refers to inhibitor, codes for repressor, and Z for beta galactosidase, and Y for permease, and gene codes for transacetylase. All three genes products in lac operon are needed for metabolism of lactose. Lactose is the substrate of beta galactosidase and it regulates switching on off of operon, so called inducer. Regulation of lac operon is regulation of enzyme synthesis by its substrate, and regulation of lac operon by repressor is negative regulation. However, lac operon is under control of positive regulation as well. Regulatory proteins can act both positively, activators, and negatively, that is repressors. Each operon has its specific operator and specific repressor. Now the next is Human Genome Project, and then after we are having the DNA fingerprinting and chapter is done. So Human Genome Project launched in 1990, a 13-year project was coordinated by US Department of Energy and National Institute of Health, and UK, Japan, France, Germany, China participated, and it was completed in 2003. Human genome has approximately 3 into 10 to the power 9 base pair, and the cost of sequencing in the beginning was US $3 per base pair, that is 9 billion US dollar. Human Genome Project lead to rapid development of a new area in biology called bioinformatics. Many non-human model organisms like bacteria, yeast, and Coinero, habitus, elegans, Drosophila plant, rice and Arabidopsis have also been sequenced. Now the methods they follow, that is the expressed sequence tags, ESTs, focused on identifying all genes that expressed as RNA. Sequence annotation, blind approach of sequencing the whole genome containing coding and non-coding sequences, needing vectors like bacterial, artificial chromosomes and yeast artificial chromosomes. Now some features of human genome. Human genome contains 3164.7 million base pair. Average gene consists of 3000 bases. Largest gene dystrophin of 2.4 million bases. Total genes estimated at 30,000. Almost 99.9% nucleotide bases exactly same in all people, and functions unknown for 50% discovered gene. Less than 2% genome codes for protein. Chromosome 1 has most genes 2968 and Y chromosome has the fewest 231. At 1.4 million locations, single base DNA differences SNPs, single nucleotide polymorphism, occur. SNPs can help in tracing human history. The fragments were sequenced using automated DNA sequencer that worked on the principle of method developed by Frederick Sanger. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, that is 22 autosomes and X and Y, to be sequenced. The next is DNA fingerprinting. 99.9% base sequence among humans is same. 0.1% differences in sequence of DNA make every individual unique in their phenotype. DNA fingerprinting involves identifying difference in repetitive DNA, a small stretch of DNA repeated many times called satellite DNA. Depending on base composition, AT or GC rich, length of segments and number of repetitive units, the satellite DNA is classified into microsatellites and mini satellites. They do not code for any proteins. They form large portion of human genome and show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue, blood, hair follicles, skin, bone, saliva, sperm shows same degree of polymorphism, they have forensic application. Polymorphism are inheritable from the parent to child, so DNA fingerprinting solves paternity disputes. The technique of DNA fingerprinting was initially developed by Alec Jeffreys, and here the steps are isolation of DNA, then the digestion by the restriction enzymes, restriction endonucleases, and then separation of DNA fragments by electrophoresis, then the blotting on nitrocellulose or nylon membrane, and then after the hybridization using the labeled VNTR probes, and then after the detection of hybridized DNA fragments by autoradiography. What is the significance of DNA fingerprinting? That VNTR are called mini satellite, a small DNA sequence arranged randomly in many copies, and the size of VNTR varies from 0.1 to 20 kb. So after hybridization with VNTR probe, the auto diagram gives many bands of differing sizes. These bands give a characteristic pattern for an individual DNA. It differs from individual to individual in a population except in monozygotic twins. The sensitivity of the technique has been increased by use of polymerase chain reaction. There are some more additional points related with the chapter, that the pressure of operon is synthesized all the time constitutively from the I gene. A very low level of expression of lac operon has to be present in the cell at the time, otherwise lactose cannot enter the cells. Lactose or allolactose is the inducer of lac operon. Glucose or galactose cannot act as inducers for lac operon. Allelic sequence variation has traditionally been described as a DNA polymorphism. If more than one variant at a locus occurs in human population with a frequency greater than 0.01. DNA fingerprinting has much wider application in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints. The hallmark of double-stranded helical structure of DNA is the hydrogen bonding between the bases from the opposite strands. So, इसी के साथ ही आपका ये इतना बड़ा सा चैप्टर कंप्लीट हो गया है फाइनली. इसमें आपको सारे पॉइंट मैंने कवर कर दिए हैं, एंड आपको कैसा लगा इसे रिवाइज करके मुझे कमेंट सेक्शन में ज़रूर शेयर करना. एंड इफ़ यू आर न्यू ऑन माय चैनल सो डू सब्सक्राइब एंड प्रेस द लाइक बटन, एंड डोंट फॉरगेट टू शेयर विद योर फ्रेंड्स. So see you soon in the next podcast. Till then, just stay tuned with me. Bye bye everyone. Good luck everyone.